Good morning, everybody who is online with this second webinar from the Health Firm project, an innovative pulse and cereal-based food fermentation for human health and sustainability diets project. And we are organizing today a webinar on nutrition and health of fermented cereals and pulses. You see Christine Verbeke and Richard Landberg, the two speakers, and Christophe Courtin, the coordinator of this project. Some household issues. First of all, as you can notice, the webinar is recorded and a link will be provided afterwards. PDFs of the presentations will also be made available. You will be notified after the webinar when these PDFs will be available and the recording will be available. There is a possibility to ask questions. There is a question box where you can address your questions. Please address the question to the speaker you want to address the question to, so that we can select questions in such a way that the questions can be asked to the speaker. Christophe Courtin will give a very short introduction on the Health Firm project. He's a professor at the Catholic University Leuven in food technology. Christophe, please, floor is yours.
Thank you, Jan, for the introduction and for introducing the webinar. It is my pleasure as coordinator of the Health Firm project to also welcome you to this second webinar. And we're very happy that we have a large crowd attending, so it's very nice to see, and it's also showing the interest of the scientific community in fermented foods and nutrition. So indeed, very short, what is Health Firm about? What is it? So Health Firm is an Horizon Europe research and innovation action project. And so last year we were awarded this project, which means that 23 partners from 11 countries can start working together on the topic of Health Firm with a nice budget that was allocated by the EU and also the Swiss government, and for a project duration of 48 months. As you can see in the lower left-hand corner of the slide, we have a very nice panel of universities, research institutes, and companies that came together to execute this project. We are really having a magnificent consortium, I can tell you. So the aim of Health Firm is actually the following. We want to have an evidence-based transition in society and industry from traditional to sustainable plant-based fermented foods and diets. And we also want to couple this to the health benefits that these fermented foods can have for customers. And we also want to design these fermented foods. So there's two important aspects in that objective. From animal-based products to plant-based products, we all know that we have to make a transition, at least in part. And also from spontaneous fermentation processes that have been gone for millennia to rationally designed fermentation processes. To do that, we need, of course, a lot of attention for microbial resources. We need to go into fermentation technology. We need to go into human health and microbiome studies, and we also have to have attention for consumer behavior, quite important. On the right-hand side, you see the emphasis that we will put on nutrition and health. So we will try to disentangle the interaction between food fermentation microbiomes, which are important in fermented foods, of course, fermented grain-based foods, because we always have to work with a matrix, human health, and also as part of that human health is the human gut microbiome. So we want to link all these elements together. And for that, we have crafted this project. And here you see the general layout of the project, the different work packages, where you see microbial resources coming back, fermentation technology coming back, the human health and microbiome, and consumer behavior, flanked by, of course, a management work package, and also work package on communication, dissemination, and exploitation. And this webinar is, of course, part of that effort to to disseminate and to communicate with you about what Health Firm is and also the broader context. So with this, I would like to end my introduction, and I will give the floor back to Jan. I wish you a very pleasant coming hour and a half with the presentations of Christine and Richard. I'm sure they will be much interest to you. So back over to you, Jan.
Thank you very much, Christophe, for the short introduction. As you have noticed, the Health Firm project considers fermentation technology and the health consequences of the consumption of it. Our very first webinar was on fermentation technology in February this year, and that was also the reason that we intended to organize a webinar on nutrition and health with some basic issues covering especially on the gastrointestinal tract. That's why we invited two speakers, also both active in the project, Christine Verbeke and Richard Landberg. And I first want to give the floor to Christine Verbeke. She's a full professor and head of the laboratory for the digestion absorption at the Catholic University in Leuven in Belgium. And her interest is research in the bacterial metabolism in the human colon, especially on how diets affect bacterial metabolism, and most importantly, what the consequences are for host health. And she uses actually the most modern technologies to do this type of research. And she has composed a presentation where she explains the basics of the human gastrointestinal tract. So how is our food digested? Christine, floor is yours. You're muted.
Indeed. Okay,
yeah.
That should be better. I will hope you can see my screen now. So thank you for the introduction, Jan, and also from my side, welcome to this webinar. And it's my task to explain to you the basic mechanisms of intestinal physiology and how our food is digested. So imagine a nice summer evening on which you are invited for a delicious barbecue. And if the barbecue has been organized by the Health Firm Consortium, it will definitely contain a lot of vegetables. Some might be fermented. The bread might be sourdough bread, or at least it will be wholemeal bread. There might be some plant-based meal replacers, and for the dessert we will definitely have fruits. So think about how you will enjoy the meal, but what I would like to explain today to you is what will happen in your body after you have ingested all these delicious foods. So we have quite some nutritional experts in the Health Firm Consortium, so the barbecue will be well balanced and you will have a nutritional high quality meal. And such a meal mainly contains of the macronutrients, carbohydrates, protein and lipids. It will also contain quite some fiber. Meals also contain salt, vitamins and minerals and polyphenols, but today these are not the topic of this webinar, so that would drive us too far. I will limit this presentation to the macronutrients and fiber. And I will explain how they are digested and fermented in our body. Now, if you look in textbooks about gastroenterology, you will find that the gastrointestinal tract has 6 main functions which are mentioned here. So of course, it all starts with ingestion of the food, and then the food is mechanically digested, which means the particle size of the food particles is reduced. So in the mouth, that occurs by chewing the food, where the process in the stomach is called churning, and in the intestine we call it segmentation of the food bolus to reduce its particle size. Then there are also 2 mechanisms that make sure that your food progresses through the gastrointestinal tract, and the first one is swallowing in the mouth, But then also in the intestine, we have a mechanism of peristalsis, which is contractions of the muscles in the wall of the intestine that make sure that your food progresses. And then, especially in the small intestine, we have the process of chemical digestions. And for that, we need the help of the pancreas and the liver. Strictly spoken, those organs do not belong to the gastrointestinal tract. We consider them as auxiliary organs because we need the pancreas, which provides enzymes for the digestion of our food, and the liver provides the bile acids that we need to solubilize the lipids. So the small intestine is the main site where the major chemical digestion occurs. And after that, the nutrients are absorbed from the small intestine into the blood. And then the undigested remnants move towards the large intestine. And we have been thinking for a long time that the large intestine was an organ that merely served to further absorb water from the undigested nutrients and prepare them for defecation. And it has only been with the advent of new molecular techniques that allowed to analyze and identify the microorganisms that are living here in our large intestine that we have seen that they ferment these undigested residues, and that this fermentation has also a large impact on our health. So the textbooks should add also fermentation as a seventh task and function of our gastrointestinal tract, I would say. So let's first have a look at the digestion of carbohydrates. So digestion of carbohydrates already starts in the mouth, where we have salivary amylase. So when you chew your foods, it gets mixed with saliva, not only to lubricate it, but also to bring it in contact with this amylase enzyme that hydrolyzes already partially the carbohydrates to smaller polysaccharides, eventually disaccharides. That amylase has a pH optimum that's somewhere between 6 and 7. So you would expect that when the food comes in the stomach, where the pH is much lower, the amylase will be inactivated. But there are indications that the pH in the stomach is not that homogeneous, and that the inactivation of the salivary amylase does not occur instantaneously, and so it keeps working at least for a limited amount of time. But then the major part of the digestion occurs in the small intestine with the help of pancreatic amylase, so coming from the pancreas, that hydrolyzes the starchy compounds further into disaccharides, and then the brush border enzymes present in the wall of the small intestine convert them to monosaccharides, and these monosaccharides can be absorbed into the blood. Non-starch polysaccharides are not hydrolyzed by amylase enzymes and make it mostly unaffected until the large intestine or the colon, where they are substrates for microbial fermentation. I will come back to that later in this presentation. Now, if you eat carbohydrates, so they are hydrolyzed to monosaccharides, glucose that is absorbed. So blood glucose will change over time. And if you eat starch that is easily digestible, for instance, if you eat white bread or sugars, then you get a kind of glucose profile like this, with a rapid rise, a rapid decline again. And sometimes the blood glucose concentrations go below the fasting state. And especially this dip in glucose concentrations may induce a new episode of hunger, may also induce sugar cravings. So with respect to obesity and type 2 diabetes, this is really not favorable, and we would like to prevent that and have foods that are that give rise to postprandial glycemic responses that look more like this curve, or even better like this one. So we prefer slow increase in blood glucose concentrations that remains then as stable as possible over a longer period of time. And you can modify the profile of your blood glucose response by changing food properties. And the first property is, for instance, the food texture. So it differs whether you eat a hard food or a soft food, because that affects the oral processing behavior, and in this way also the exposure to salivary amylase. Another one is the quality of the starch. So crystalline starch will be less easily or less rapidly fermentable than amorphous starch. The botanical structure can also have an impact, and especially whether there are still intact cell walls present in the food, because that limits the accessibility of the digestive enzymes to the starch and will at least delay the digestion of the starch and maybe also reduce it. And then also some types of dietary fiber can have an effect, and we think especially at the viscous fibers such as beta-glucans, because they increase the viscosity of the intestinal content, and then in this way reduce the diffusion of nutrients to the intestinal wall, and so delay or prevent absorption of them. But it's not only the food properties that influence the postprandial glycemic response. There are also characteristics of the individual itself. And there is more and more evidence that if you give the same food to different people, that the postprandial glucose response can vary quite a lot. And this is an example here. So here we gave, well, it's not our results, it's a study from the literature. So they gave exactly the same food to different participants, and you have a huge variety in glycemic responses. So based on this observation at the Weizmann's Institute in Israel, they tried to predict glucose response using a model. So they constructed a model. The model contains 72 features. Some of them are meal-related features, such as, for instance, the amount of carbohydrates in the meal context features, such as timing of the eating, blood tests like fasting insulin concentrations, personal features like HbMI, continuous glucose monitoring features, and also microbiome features. So a lot of different, very different parameters that were all combined into one model. And using that model, they really could predict which people would respond with higher glucose concentrations in the plasma to these foods. So here you see the predictive model. If it would work, if you would predict the glucose response only on caloric content, you have a curve like the red one. And you have to know so that the one-to-one, the bisectrix of this curve is actually arbitrary. So this does not, this is not very different from a straight line here, which means that it does not have hardly any predictive value. If you use the carbohydrate content of a meal to predict the glucose response, it's only slightly better. But if they used the model that has all those 72 features, it was much better to predict which people would be in the highest 50% percentile of the measured glucose responses. And if they take the 90% of well, actually the 10% of highest glucose responders, that was even much better. Now, based on this model, they did another study in which they personalized the diet of more than 200 people with pre-diabetes. Half of the people received a control diet, which was the Mediterranean diet. The Mediterranean diet is actually considered as one of the best diets that you can have. And the intervention diet was the personalized diet in which the meals got a score based on this model with the 72 features. And the participants received the meals for 6 months, and all of them had a sensor that continuously measured their glucose response. And then they saw that both after 3 months and after 6 months, the time then in which the participants had glucose concentrations above 140 milligram per deciliter, which is considered as a kind of threshold for negative effects related to diabetes, that this time significantly decreased. It was also lower in the control group on the Mediterranean diet, indicating that this is also a good diet, but the effect was much more pronounced in the personalized group. Also, the percentage of glycated hemoglobin, so the hemoglobin A1C, which is considered as a marker for long-term glucose exposure, was significantly better in the personalized diet group. So, of course, you have to realize that the people did not eat the same foods, did not eat the same amount of carbohydrates, but I only show you this to show that there is a huge difference in how people react to meals, and you can try to modulate that. Now, last month there was a new study in which they used the same diet, but now combined with caloric restriction. So the diets, both the control diet and the personalized diet, induced a calorie deficit of 500 kilocalories, and here you do not see an effect anymore. So both diets decreased a little bit in glucose excursions and hemoglobin A1C, but it was not significant, and the effect that you saw was considered as clinically not relevant by the authors. And also there was no difference between the two, between the control diet and the personalized diet. Again, not everyone had the same amount of carbohydrates, but just indicating that by modulating the food is not the only thing that that influences the food properties are not the only thing that influences this postprandial glucose responses to food. And actually we do not completely understand what the best, or what the parameters and the characteristics of the individuals are that have an effect. Okay, let's continue with the digestion of protein. Digestion of protein starts only in the stomach with pepsine hydrolyzing part of the proteins ready to polypeptides, but also here the majority of digestion occurs in the small intestine with enzymes secreted by the pancreas, trypsin, chymotrypsin, carboxypeptidases, and then we also need peptidases to convert smaller peptides further to amino acids that can be absorbed. There are sometimes a little bit of protein can escape digestion in the small intestine, and so then reach the colon, where also these proteins are substrates for microbial fermentation. There are some differences. Well, actually, there are two main differences between plant-based proteins and animal proteins. And the first difference has to do with the amino acid composition. So you might know that there are 9 essential amino acids. You see them listed here. And those essential amino acids, it means that we cannot synthesize them in our body ourselves, but we have to take them up via the diet. Animal protein contains all of these essential amino acids in appropriate proportions, but that is not the case for all plant-based proteins. And for instance, cereals are deficient in lysine, the lysine amino acids, and legumes are often deficient in sulfur-containing amino acids. It is not too much of a problem, because if you combine in your meal cereals and legumes and make clever combinations of food compounds, also with plant-based with the plant-based diet, you can make sure that you have all the essential amino acids in the right proportions. A second difference between plant-based and animal proteins is their digestibility. Animal proteins are very well digested, 90 to 95%, and digestibility of plant-based proteins is often a bit lower and is estimated 75, 80, 85%, something like that. The lower digestibility is due to the presence of what we call anti-nutritional factors, such as protease inhibitors, lectins, phytates, fibers, and polyphenols that all can decrease digestibility of the protein. You might discuss whether we have to call all of them anti-nutritional factors, because, for instance, fibers and polyphenols also have definitely clear nutritional benefits. but here in this context, it only means that they decrease the digestibility of the proteins. There are also solutions for that, and you can decrease the concentration of these anti-nutritional factors, and one of them is fermentation of your food. By fermentation can improve the protein digestibility, and I just show you here a number of examples. The upper light gray bar is always the unfermented food, and then the darker gray bar is fermented with different types of fermentation. I would say that here the difference in beans is quite spectacular. And you also see that for instance, fermented soybean reaches a digestibility that is quite comparable to that of animal protein. So you can really change that, and so that also this lower fermentability of plant proteins is not really a big problem. Digestion of lipids also starts in the mouth with lingual lipase. There is also gastric lipase in the stomach that accounts for about 10% of the digestion of the lipids. And also here we rely mainly on enzymes from the pancreas to digest quasi all lipids in the small intestine up to fatty acids. And then the bile salts from the liver are required to solubilize the fatty acids and make sure that they can be absorbed. There is not so much known about fermentation of fatty acids in the colon, which is probably due to the fact that the colon is largely anaerobic, so there's no oxygen, which makes that fatty acids cannot be oxidized very well. And we assume that most of the fatty acids that are not digested in the small intestine are just excreted in the feces. So that means that if we consider the undigested nutrients that reach the colon, we estimate that the amount of protein is about 12 to 18 grams per day. The amount of carbohydrate has been estimated between 5 and 50 grams per day, but that can be very, very much depending on the diet that you have, which is clear here from this figure. So here in the y-axis, you see the loss of carbohydrate. So the amount of carbohydrate that spill over into the large intestine per food containing 80 grams of carbohydrate, but different amounts of fiber. And so, for instance, the beans, you can have quite a lot of carbohydrate losses, also without bran, for instance. And so there is a large variability, and depending on the diet, this is a factor that can really modulate. The digestion of lipids is quite efficient, and normally assuming a daily intake of 100 grams of fat per day, normally you expect not more than 7 grams of fat excreted in the feces. Now, 100 trillion, so 1 and 14 zeros, that is the estimated number of bacteria that lives in our large intestine, and that can use all these undigested nutrients. Now, before we continue, I would like to say a word about the terminology, because some people talk about microflora, some about microbiota, some about microbiome, and what is it now? I would say microflora is an old term that has become a bit obsolete and that I would not recommend to use, because it's about microorganisms, not about flowers. So yeah, that is not the best option. But then microbiota and microbiome are often used interchangeably, while actually they are two different things. And when we talk about microbiota, it's really the microorganisms themselves. So in the large intestine, we have mainly bacteria, but there are also some archaea, protists, fungi, algae, and they are all considered in the term microbiota. When we talk about microbiome, you can talk about it in strict definition. And when you use the strict definition, it's actually the genetic material, so the DNA and RNA of those microbiota. Part of this DNA and RNA, mainly the biggest part, is within the microbial structural elements themselves. But there are also elements that are outside the microbiota. For instance, the mobile genetic elements that transfer resistance genes from one organisms to another, or the viruses or the phage DNA, which can occur in the environment, but they are also included in the microbiome in the strict definition. However, some also use the microbiome in a much broader definition, and then it means the microbiota together with what we call the theater of activity. So, and that is the whole thing of all microbial structural elements, including protein, peptides, lipids, polysaccharides, the nucleic acid structures, also the metabolites that have been produced by the microbiota. Some of them can be signaling molecules, some of them can be toxins. There are also just organic molecules, and also the environmental conditions like oxygen concentration of pH are all included in this whole environmental theater of activity, let's call it like that. So you can use microbiome in a strict or in a broad definition. When we talk about microbiota, it's about microorganisms themselves. Now, we and our microbiota live in a symbiotic relationship, which means that both parties have benefits of living together and that actually we need each other. So what we provide to our microbiota is a continuous supply of nutrients and an anaerobic environment. So there is no oxygen in the large intestine, which is important because most of these bacteria do not tolerate oxygen. It's also nicely warm. So for the bacteria, it's really the ideal place to live. But we also have benefits from our bacteria because they help to digest our food and they also interact with our immune system. Now, for today, I will only talk about how these bacteria help to digest our foods. And then it's clear that there are regional differences in the colonic fermentation. And when we look at the proximal colon, so here is the site where the nutrients from the small intestine enter the colon. This is the place where there is ample of substrates and where the bacteria can choose what they want to ferment. And if bacteria have the choice, they have a preference for carbohydrates because fermentation of carbohydrates is energetically more favorable for them. And it's only if you progress through the colon where the carbohydrates get depleted that protein fermentation becomes more important. So here in the distal colon, we have more protein fermentation. Now, besides the microbiota, diet and transit are the main modulators of the fermentation. So you can really influence these processes in your own large intestine, mainly by varying the diet. And varying the diet then means varying the proportion of carbohydrates to protein that arrives in the colon. And actually, it is varying the amount of fiber that you eat. Now, diet, transit, microbiota composition are not independent parameters. They are related to each other in the sense that if you eat more fiber, you will also affect your transit and you will affect your microbiota composition, which will become more diverse and more give more carbohydrate fermentation. Now, the process of carbohydrate fermentation is a process that has been studied quite well. We know quite good how that occurs, and it's not all the intermediates are not mentioned in the schemes, but if you come from carbohydrates, polysaccharides are first hydrolyzed to monosaccharides that then are converted to pyruvate. So pyruvate is the central molecule in this cascade of reactions, and pyruvate can be converted to propionate via succinate, or it can be converted to acetyl-CoA that further goes to acetate, and acetate with acetyl-CoA can go to butyrate. There are also 3 reactions that produce hydrogen. Hydrogen is a kind of side product of carbohydrate fermentation. It's not harmful, but it can be annoying and cumbersome, but because it can give can induce flatulence. No, accumulation of hydrogen would in the colon would very rapidly restrict the fermentation for thermodynamic reasons. So it is important that the hydrogen is also removed from the large intestine. And there are 3 types of bacteria that can do that. So we have the methanogens that convert the hydrogen into methane, the reductive acetogens that convert hydrogen into acetic acid, and the sulfate reducing bacteria that use the hydrogen to for the reduction of sulfate. Now, these propionate, acetate, and butyrate are what we call the short-chain fatty acids, and we think that these are very important molecules. You see their chemical structure here. These are very simple molecules, very small molecules, but they are the main anions in the colon. They are produced from dietary fiber in amounts of about 500 to 600 millimoles per day, which is quite a lot. And we think that they may be the link that connects the diet, the microbiota, and the host. Short-chain fatty acids have local effects already in the gut, and one of them is the fact that they affect the pH. So if this scheme gives you the pH throughout the gastrointestinal tract. So at the end of the small intestine, the pH is about 7.5, something like that. And then when you go to the large intestine, to the colon, it immediately drops from 7.5 to about 5.56. And that is only due to the production of these short-chain fatty acids. Later on in the large intestine, the pH already increases again to about 6.8 to 7 in the fecal samples. Now, this lower pH here is actually beneficial for our health because it's on the one side, it inhibits proteases, so it inhibits the enzymes involved in protein fermentation. And on the other hand, it inactivates some pathogens. For instance, Salmonella, when you get infected with Salmonella does not tolerate these low pH and will be quickly inactivated if the pH in your proximal colon is low enough. In addition, short-chain fatty acids are also the main energy source for our colonocytes. So they are, when they are produced in the colonic lumen, they are rapidly absorbed by the colonocytes, and within these cells, they serve as an energy source to generate ATP for the cells. So it's important also for the health of your colonic mucosa that you eat sufficient amounts of fiber or fermentable fiber and that you generate enough short-chain fatty acids here. The fraction that is not oxidized in the colonocytes goes via the port of circulation to the liver and finally ends up in the systemic circulation. So the fact that short-chain fatty acids also reach the systemic circulation means that they can affect other organs. And the first organ that they reach is the liver. And we think that short-chain fatty acids also affect lipid metabolism, for instance, in the liver, but they can also affect the pancreas, the brain, the adipocytes. So they can also have impact on organs on a distance. Protein fermentation also results in a wide variety of metabolites. So protein in the colon is either originate either from malabsorbed dietary protein or from endogenous proteins, such as, for instance, the pancreatic secretions contains proteins that make it into the large intestine and also get fermented by the microbiota. Part of these proteins is used for proteolysis by the microbiota themselves, but the majority is fermented into a range of metabolites. There are nitrogen-containing metabolites, aromatic compounds, sulfur compounds, acids. So protein fermentation also gives rise to some short-chain fatty acids, but clearly lower amounts than carbohydrate fermentation. And there are also branched-chain fatty acids. And then again, we have some gases. Hydrogen, CO2, and methane are also produced with carbohydrate fermentation, but hydrogen sulfide is typical for protein fermentation. Now, all these products need to be disposed, and there are different possibilities. So they can be excreted in feces, so the gases can be excreted in bread, so then you get rid of them, and okay, it's fine. they can be utilized by the microbiota, but several of these fermentation metabolites are also absorbed into the systemic circulation. Some are already detoxified in the colonic epithelium, and some are also further metabolized by human enzymes. And let's have a closer look at the metabolites of aromatic amino acids, because they might be important for our health. So there are 3 aromatic amino acids, tyrosine, phenylalanine, and tryptophan, and they can be fermented by the bacteria into a variety of metabolites. But especially these ones, so phenol, cresol, and indole, have been implicated in, have been considered as detrimental for our health, and they have been mainly implicated as uremic toxins. So they are produced in our large intestine, and they are quite extensively absorbed by the colonic mucosa, and then detoxified by sulfatation. So they are converted in paracrysyl sulfate, indoxyl sulfate, either in the mucosa or in the liver. For healthy people with a normal kidney function, it's not really a problem, and we will quite quickly excrete these metabolites via the kidneys into the urine. But in patients with kidney dysfunction, it becomes a problem, because they do not excrete them into the urine, and so they accumulate those compounds in their blood. And the main problem is that these compounds are highly protein bound, so they bind to albumin, which means that they also cannot be eliminated with dialysis techniques. And it has been shown that especially paracrysyl sulfate and indoxyl sulfate are involved in cardiovascular toxicity. And I don't know, maybe you don't know, but people with kidney disease most often do not die from kidney failure, but they die from cardiovascular events. And we think that these protein fermentation metabolites are involved in this cardiovascular toxicity. In contrast, tryptophan metabolites, which is then the indole, but also indole propionic acid, indole acetic acid, have been also now more recently been thought that they are involved in intestinal barrier function and in immune function, and that they would improve those functions. So they might also have beneficial effects, but it seems to be really dependent on where these metabolites are, whether they are in the gut or whether they are in the systemic circulation. So this is something that has been shown at this moment mainly in vitro studies and in animal studies, but that we still have to translate to humans, and that at this moment we only start to understand what the impact is of all those metabolites on our health. So I just wanted to give you an impression of the wide variety of metabolites that is produced by the bacteria, and then all of them, well, many of them can affect our host health, but that is something that we still do not completely understand. And a main problem in that is that all those results mainly come from in vitro and animal studies, and it's very difficult to study that in humans because we cannot sample in the terminal, in the small intestine, for instance, or in the colon. We cannot take samples from the colonic content in the proximal colon. We just cannot treat those sites. And with that respect, I wanted to draw your attention to a very exciting paper that has been published 2 weeks ago in Nature. And at least I find it very exciting. And where they used a new device, so 4 different capsules, and the capsules were coated with different coatings that dissolve at specific places in the small intestine and in the colon. And so the dissolution of the coating was based on pH and time differences. The capsules contain a kind of small bag that is folded into the capsule, and that is vacuum. And so when the capsule dissolves, the bag unfolds, and it sucks intestinal fluid into the bag, and then you have to retrieve the bags in the stools. The bags contain, like you see them here, about 400 microliter of intestinal fluid that then has been analyzed. So this is a very novel technique that allows to really sample in the gastrointestinal tract and allows to study what happens at the location. And what they found was that there are very big differences in microbial community depending on the location, that human protein abundance differed between stool and intestinal samples, so that what we measure in humans in stool samples is definitely not what happens in the intestine. And also the bile acid profiles were very different along the gastrointestinal tract compared to what we have measured for so long in stools, because that was the only matrix that we had available. So that brings me to the conclusions of this presentation. And I think that we have to say that in healthy conditions, our gastrointestinal tract really works as a well-oiled machine. So there are different processes that are related to each other and that well coordinated and work fine, so that overall the digestion of macronutrients is very efficient. We do not need to improve that. I think we even want to decrease the digestibility, especially of rapidly digestible carbohydrates, where we would like to have a more gradual digestion and release of glucose in the blood. Fermentation of undigested nutrients yields a wide variety of metabolites that can affect the host, and I emphasized it already many times, several times. We are really only at the start of understanding what this means for our health. And with novel technologies to sample different locations within the gastrointestinal tract may help and further increase our knowledge about these fermentation products. And with that, I would like to thank you for your attention.
Thank you very much, Christine, for this very, very nice introduction on gastrointestinal physiology, including all those new technologies to measure indeed over time what's happening in our gastrointestinal tract. It's very intriguing what's happening, and definitely we have measured. We don't know actually what's happening there in that long tube that goes from mouth to the bottom. We have Rickard Landberg, who is going to introduce us to some more details with respect to the evidence that we have on the role of fermented foods in health. And Rickard is a full professor, head of the Division of Food and Nutrition Sciences at the Chalmers University of Technology. He also is part of the Health Firm project, and he studies the preventive role of plant-based tools using observational and intervention studies. He leads several very interesting studies, and he's going to introduce you on fermented foods for health. What are the evidences? Rickard, floor is yours. You're muted. Can you turn on your mic?
Thank you very much for that kind introduction. Yes, Okay, so fermented foods for health, what are the evidences? I need to start to say that I have been working on this in the Health Firm project together also with my postdoc, Elise Nordin. I think we need to first start to put the fermented foods in a little bit wider context. And nowadays, there is a lot of discussions of how biotechnology can be utilized in the protein shift era, where we try to go from more meat-based diets into more plant-based diets. And then the concept of microbial foods, which is a little bit wider concept, often come up. And in this concept, it's basically three different parts, where fermented foods is representing one of these three parts. Fermented foods refers to traditionally produced ones from grains, seeds, fruits and vegetables, but also dairy, meat, fish, and so on, where we have controlled transformation by microorganisms. But we also have microbial biomass, where we are producing from a sugar source. Typically, it can be a side stream or any type of sugar source, where we are producing different microbial biomasses that are utilized also for food purposes. And we also have the cell factory system, where we have microorganisms that can be highly tailored or genetically modified in order to produce specific components that we are using as ingredients typically in foods. But today I'm going to focus most on the fermented foods. All of these, as I mentioned before, are essential, I think, for the transition that we are going to make into more sustainable and also healthy diet. So all of them have a role in this transition. So what about fermented foods? Fermented foods means foods that are made through desired microbial growth and enzymatic conversion of food components. And as I mentioned, it's a wide diversity of different types of foods. Fermented foods have been consumed for a very long time in history, and it has its roots in Asia. Almost 1/3 of the foods that we are consuming are in one way or the other fermented. And we have more than 3,500 foods, different types of foods that are belonging to this group. So it's an extremely wide group. Why do we ferment foods? I mean, back in history, it was very much about stability and making foods safe and making also food security. This was a very important food security issue, not the least for us here up in the north, to ensure that we had foods on the table also in the early spring or in the winter. So it's very much about stabilizing the foods and provide a context where the food is safe to consume. So typically adding microorganisms that will lower pH and thereby preserving the food items from microbial spoilage. It's also about sensory. We like to eat and drink fermented foods and beverages due to the great diversity of taste and flavor components that are formed in these products. And of course, also the perceived health benefits that has been even more important lately. And I write perceived health benefits here, and that's something I will revolve around because very much has been thought about the health benefits or potential health benefits, but the evidences are actually quite scarce, and particularly for the new type of fermented foods and the plant-based fermented foods, whereas most evidences and most studies in the history has been more on the dairy foods, for instance. So this is very much a black spot on the map when it comes to evidences for health. But not the least, still, this is discussed a lot in popular science and in media, not the least, where you get the impression where the consumer get the very clear impression that fermented foods is healthy and it's good for you to consume fermented foods. But the evidences are, as I said, not lined up to that claims. So how does it look like? I mean, when it comes to recommendations and claims? Well, despite the many potential benefits of fermented foods, the recommended consumption has not yet been widely translated into global inclusion in food guidelines. We can see that some fermented foods are in the national guidelines of several countries, but then they are not emphasized as being fermented. It's more like food groups that represent fermented food groups that are mentioned for different reasons, and not specifically because they are fermented. And this is, of course, partly due to the lack of firm evidences for these foods. One exception is in Asia. That was quite astonishing that this has not been more widely adopted in Asia, where everything started and where there is a lot of emphasis and has been for many years on fermented, also plant-based foods such as soy products. But there is one exemption, and that is in India, where they are even stressing in the official dietary guidelines, even for women, that pregnant women, they should eat more whole grains, sprouted grams, and fermented foods. So their fermented foods is actually mentioned explicitly. When it comes to health claims in Europe, we have one health claim for beneficial microbes that have been approved, and that's about yogurt and that improves lactose tolerance. And that's quite thin regarding the huge efforts that have made over the years in research and development in this area. I think this picture shows a little bit one of the reasons for the challenges that we have in this area. This is just a description of different microbial content in different commonly fermented foods. So you can see in kefir, kombucha, sauerkraut, tempeh, and so on. You can see that in some of them there is a spontaneous fermentation process, but most of them have used different types of starter cultures. And you can see to the right, there's an enormous variety in different microorganisms that are present in these foods and that are dominating in these foods. And of course, different microorganisms means a huge diversity when it comes to metabolic effects, metabolic activity, enzyme activity, generation of different bioactive compounds, possibility to degrade and convert food constituents. All these things all these processes that we think are of great importance for the effects that we are later measuring related to health. So of course, with this diversity, it's very difficult to pinpoint specific mechanisms and actions that might be at play for health benefits and even for nutritional benefits. So it's a very complex we are working with a very complex material, and that's why it's also very complex to disentangle the role of these specific foods and microorganisms and the molecules that they are generating. So very little is actually known about the health effects or substantiated by evidence in trials. Many of the studies that have been conducted are observational studies, and the few interventions that have been conducted are very often they are very small, they are very selective groups, and they are of quite poor quality in most cases. The example is perhaps dairy products, fermented dairy products, which represent the group of foods that have been investigated the most. And when we are thinking about fermented foods, we should of course keep in mind, particularly when we are thinking about the plant-based fermented foods, we should keep in mind the concept of prebiotics and probiotics, and this has been around for quite some long time, and I guess most of you are very familiar with this concept. But we also have the concept of postbiotics, which is a little bit entered the scene a little bit later. And postbiotics means basically that postbiotics means a preparation of inanimate microorganisms and or their components that confers a health benefits to the host. So it's actually not a live microorganism. It's dead microorganisms or parts of dead microorganisms, content of or bacterial lysates from this dead microorganism. And it might also be metabolites from these microorganisms. So the resulting products from the activity of these microorganisms once they were alive. So this concept is a little bit broad, but it deals with the material basically that comes from the organisms, and that to be regarded as a postbiotic, it must also have a contribute to the beneficial health effect that is at study. I think this was something that Christina was alluding a little bit into. I think whatever we are feeding, I mean, also including the fermented foods, there is a lot of metabolites produced or potentially bioactive compounds produced by both in the food material itself, but not the least when this food reaches the intestine. And I think it is very important to keep those things in mind. So we should not only focus on the bacteria itself or the residues of the bacteria, but we should really focus on what are the metabolites formed when these fermented foods reach the colon, because these metabolites will, as Christina also mentioned, potentially be very important mediators of the effects, both of the foods that we see and also of the gut microbiota and the alterations of the activity of the gut microbiota that these foods might generate. So it's important to keep these molecules in mind. When we look into the health effects of fermented foods, I mentioned yogurt, I mentioned dairy products, and there have been studies conducted, some quite recent reviews and some meta-analysis looking into observational and also the few, not in the area of the dairy foods, but in fermented foods in general, in intervention trials. And one can conclude to say that there are associations between yogurt intake and reduced risk of several chronic diseases. This is just one example of relative risk estimates for the increased consumption of yogurt and the impact it has on mortality, both all-cause mortality and CVD-related mortality, for instance, and cancer mortality. And one can say that there are associations with some types of cancer. Breast cancer and colon cancer is what has been most commonly studied. There is most consistent and strongest association, inverse association with type 2 diabetes. So the more yogurt you consume, the lower the risk of developing type 2 diabetes or dying for cardiovascular disease, where type 2 diabetes is one cause. It's going down. It's also studies showing improved weight management, also effects on inflammatory markers and bone health, of course, and also gastrointestinal health, which is probably the strongest effect or endpoint where we see an effect of these type of foods. There was another meta-analysis, or not a meta-analysis, a review article that went through the different studies, basically investigating the effects of these foods, of yogurts or dairy foods. And you can see very clearly here that, I'm sorry for the quality of this picture, but you can see that in this box you see the studies which show a favorable outcome, and at the far end you see those with an unfavorable outcome, and then you have those with neutral effect in the middle. And you see that there is a tendency, at least for body weight and for diabetes, of course, you see very clearly 7 studies with an effect, whereas 1 without any effect, that there is a tendency of an effect in these studies. Although I think the authors made a quite too bolded statements where they say that fermented milk, that there are consistent associations between fermented milk consumption and reduced risk of breast cancer and colorectal cancer, type 2 diabetes, improved weight management, and improved cardiovascular, bone, and GI health. I think it's a little bit too strongly stated, but it seems that there is something in there, but larger and more and better studies are desperately needed, I would say, even for something such as established as dairy products. These are some examples also of other type of products. This is kefir on gastrointestinal health, and there have been investigations in different trials on different types of measures of gastrointestinal health, and some studies show also here there is a tendency that the studies show a beneficial effect, but it's not strong, and it's mixed across different studies, as often the case. And it's the same when looking into sauerkraut, soy product, and kimchi, where the number of studies are even fewer. There are effects on stimulation of specific bacteria, bifidobacteria, for instance, and lactobacilli, and so on, which we have seen is positive for health or for nutritional aspect for some aspects. It's also studies showing eradication of Helicobacter pylori, which is the stomach ulcer bacteria, but these are small studies and we should be precautious when we interpret these findings. I think one aspect which is very important is, which has been very much neglected, I think, in the whole transition of the green transition, it's actually the role of cereals in the whole transition. We know that cereals is the largest energy source in the diet worldwide. It's also the largest source of plant-based proteins in the diet worldwide, and that's very much forgotten, I think, in this green protein shift that we are in right now. And it's also, of course, the main source of carbohydrates and dietary fibers. So it's a very important staple food. And of course, we can treat our cereals in different ways. We can have whole grains, which is of course preferred when we have higher fiber content and also more bioactives and so on. But we can also produce fermented products such as sourdough breads, for instance. And there have been investigations also into the role of sourdough breads when it comes to metabolic effects in more short-term studies. And here is a summary of those. And we also see that there are some studies showing benefits and some not, of course, as usual. And the classical mechanisms for why sourdough should be positive has been attributed mainly to glycemia, where there have been postulated positive effects of the acid, the organic acid produced, that causes delayed gastric emptying rate, and that positively affect glycemia and so on. But early studies that have been in that field is very much model studies and with very unrealistic contents of acids and so on. So the question is, in a real life setting, how practical these type of mechanisms are, how relevant they are in real time, in real life situations. Whereas, of course, bioactive compounds that are generated and liberated might be more relevant for the health effects. I will show you some, I will come back to that. I will show you some results on metabolic effect related to fermentation of cereals that we have done in our own studies. But first, just to summarize, that we have a very limited evidence on the effectiveness of most fermented foods on gastrointestinal health, and also I would say the other cardiometabolic diseases also overall, and cancer definitely. I wouldn't dare to say anything about cancer, actually. I think it's a very limited, those studies that have been conducted, and two bolded statements in the literature in relation to what is actually there. Majority of studies are of very low quality. They are small, short duration, and poorly characterized material. So then I turn a little bit to the metabolic effects of fermented versus non-fermented foods. And we have done this with different types of rye-based foods in several projects. So in this particular project, together with SLU, some years ago, we investigated whether there was a difference between fermented and unfermented bread when it comes to appetite, or basically satiety, and also glycemia. And this was in a postprandial setting. And what we started to do, which is not very often done, is actually the thorough characterization of this material that we used in our nutritional study. So we stained the different fiber components, and we compared unfermented crisp bread versus fermented crisp bread versus also refined wheat control. And what you could see, what struck us very clearly here, is that we have a much more loose structure. We have a much more degraded structure in the fermented crisp bread compared to in the unfermented crisp bread. And we think this could have potentially even negative effects, metabolic effects of the fermented foods, because that will make blood glucose to raise faster and more in the same way as for the wheat bread compared to the other unfermented bread, for instance. So we were thinking that, hypothesizing that perhaps even fermentation can negatively affect the metabolic effects compared to unfermented material. And here are the results when it comes to satiety. So this is on the y-axis, you see the satiety, and then you see the time. So they were provided a breakfast with these products, and then we asked them every half an hour how full they felt at that time. And you can see that they are fuller when they consume unfermented rye compared to the wheat, and also a little bit fuller than the fermented rye, but there was not a big difference. There was not a significant difference between the fermented and unfermented. So rye makes them to be more full in this case. When we look into glycemia, we don't see an effect, but we see a clear effect on the insulin level, and that is something also which relates to the structure of rye, and which can be illustrated by the first figure I showed. Because in the rye, and the non-fermented rye product, it's less disintegrated. Glucose is rushing into the system slowlier, and which puts a lower demand per time unit to excrete insulin, and that's why the insulin levels are lower in this case. So we could see that rye, of course, and also fermented, non-fermented rye was better than the fermented rye in this case, due to the structural more integrate material that we had with the unfermented rye. We also wanted to compare sourdough fermented then versus yeast fermented to see if that was comprising a difference. We also added viscosity measurements here, so we also analyzed the viscosity, and we saw also very clear difference in viscosity between these different foods. So the unfermented rye was much more viscous compared to the fermented product. So this is the sourdough fermented, and this is the yeast fermented, and that is also partly explaining potentially the effect that we see on glycemia. In analogy with viscous forming beta glucans, we also have viscosity in this case, and the effects could be due to similar reasons as in that case. We could also see clearly that there was an around 20% difference in hunger in this case, but it was also reciprocally in fullness. So we have a 20% beneficial effect on appetite, basically on the sourdough fermented rye compared to the other. We didn't see a difference in the glucose, but we also saw a corresponding difference in insulin as in the other study. Then we wanted to conduct a study to elucidate whether the amount of rye versus the amount of sourdough had any implications for appetite. So we conducted a factorial experiment. In this time it was 23 subjects, healthy subjects, and it was also soft bread that we used in this case. So we had 2 different levels of rye content, and we have 2 different levels of sourdough, and then we have a center point, and then we also have a refined wheat control products. So 30%, 35 to 48%. So the range was rather narrow for the rye content, whereas 9 to 51% sourdough. So that was a wide range of differences in sourdough that we had. So this is basically just to show you the design. So here we have the rye flour, the percent rye flour of total flour, the percent sourdough of the total dough. And here you can see the lactic acid was different, for instance. So we have basically acid equivalents, and the pH differed as we expected, of course, across these different levels of sourdough in the breads. And they were very sour. Those with a lot of sourdough were extremely sour. So this is really a model system. When we looked into fullness, we could see very clearly that these were the breads where we had a statistically significant difference. The difference is not very large, but it's significant. And it's totally dependent on the amount of rye. So you see that even with low sourdough and high sourdough and medium, it doesn't make any difference in fullness. So they feel as full irrespective of the sourdough content. That was also verified when we did an ad libitum energy measurements in a lunch meal after they conducted this controlled appetite rating. So they had an ad libitum lunch, and then we checked how much they were eating, and we could see very clearly that those who had more rye, they ate less. But sourdough content did not play any role at all in this. So we concluded that there was no difference in the sourdough amount that we added on appetite, whereas there seems to be an effect on rye, and that is consistent with what we saw before. So the conclusion of this was that, if anything, sourdough doesn't seem to have a big effect here on appetite and on glycemia and so on. It might even be a negative impact of having fermented material compared to unfermented material when it comes to quick metabolic responses. And that is something that is not very often discussed. We tend to talk about the positive effects of potential bioactives that are formed or liberated and so. But that's important to keep in mind, I think, for future developments. When we look in general, what are the mechanisms behind fermented foods when it comes to cardiometabolic health, which is a main focus of our research? There are different proposed mechanisms for how live microorganisms, probiotics, or inanimate microorganisms, postbiotics, or the metabolic products of these fermentation processes affect health. And we have heard already short-chain fatty acids seem to be very important mediators, not only for local effects in the intestine and immune stimulation and so on, but also on cardiometabolic risk factors. We can affect the gut microbiota composition. It's a question mark to what extent, the composition is of major importance, rather the activity is probably more important. Several of these, both the postbiotics and probiotics, can modulate or affect the interaction with the gut epithelia and affect the barrier function. We can have bioactive peptides form, for instance, during the fermentation process, and a lot of other metabolites. I just listed a few at the beginning. And we can, of course, also affect nutrient bioavailability. I think this is one of the most important aspects when it comes to utilization of fermentation, not least in the green protein shift, that we reduce the levels of antinutrients and make minerals and other components more bioavailable. And not only focusing on the protein bioavailability, but particularly the bioavailability of other nutrients and so on that are more minor. And these molecules and processes will affect energy homeostasis. It can affect obesity, insulin resistance, inflammation, and so on, which are important mediators or risk factors of cardiometabolic diseases at the end. I mentioned that interventions are very few when it comes to fermented foods, and that most of them have been evaluating dairy products and also other products where there is just one product at the time that has been tested. I think this, I want to draw the attention to this study, which was very interesting, that came 2 years ago by the Sonnenberg lab in US. And this was a study where they wanted, it was a small study, but it was quite well thought through and quite comprehensive. And in this study, they wanted really to evaluate the health effects of fermented foods versus plant-based foods. So they compared a portfolio diet, one can say, of different types of fermented foods compared to a high fiber plant-based diet, basically, or very rich in plant-based material, in order to elucidate differential health effects mediated then by fermentation or plant-based food. So they recruited 18 subjects to this study, and they had a run-in phase where they ramped up the intervention products. Most of these products where they advised the subjects to consume these products, so compliance might have been an issue in this trial. So they had 3 weeks to ramp up, and then they have 6 weeks to do the intervention, and then they had another 3 weeks at the end where subjects could choose the amount that they were consuming. So in the plant-based diet, they added 20 grams of fiber, so they had more than 45 grams of fiber a day in that group. And in the fermented arm, they had 6 servings per day. I should mention that these products were not matched, so they didn't use like fermented plant-based foods and then unfermented plant-based foods, which would have been much more elegant in relation to understanding the health effects at the end. But that was not done in this particular study. And the primary endpoint here was the cytokine response score. So they basically analyzed a lot of compounds related to inflammation, in this case on cytokines, different types of cytokines, and formed a score. And they wanted to see a difference in this score comparing the two diets and also compared to baseline. And secondary measurements were basically everything else. So microbiota, short chain fatty acids, a wide set of inflammatory biomarkers, lipid profiles, and so on. And what they found was that they didn't see any effect on the score, but they saw clear effects on the different inflammatory biomarkers, both with the plant-based group, but also to some degree with the fermented food group. They could also see very clearly that the fermented food group had a much more diverse microbiota. So their diversity of their microbiome increased in that group. And they could also form different clusters depending on how they grouped or they analyzed several cytokines and inflammatory markers, and there they could see that individuals responded, particularly on the plant-based diet, differently. So they could form 3 different response clusters, which were also related to some degree to the changes in microbiota in this study. So it was a very interesting study to see very clearly that in the high fiber diet, in particular, they increased the microbial functionality towards more carbohydrate degrading enzyme activities, formation of more short chain fatty acids, and establishments of personalized immune responses across these 3 different clusters I mentioned. whereas in the high fermented food groups, they increased the microbiome diversity and decreased inflammatory signals and activity. So this was the sort of main outcomes of this study. And of course, now it would be interesting to follow up on this data that they presented in the trial. And this is exactly what we are going to do now in the HealthFirm project. So we are right now designing a study which will soon be available in the clinicaltrial.gov, so you will be able to look at the protocol in more detail. But it's a fermented plant-based portfolio diet for health, for metabolic health. We call it Ferm-D Health study. And the primary aim here is to find a difference in inflammatory biomarkers. And we will have secondary aim: lipid profile, insulin, blood pressure, glycemic variability, microbiota, metabolome, and we will also do some stratification. I will come back to that. So the idea is to have a randomized control crossover trial with 100 people with signs of metabolic syndrome that will have 3 different diets in a crossover fashion. So here we will have a fermented plant-based food diet portfolio. So we have a combination of different fermented plant-based foods, and we will compare them to corresponding foods, plant-based foods that have not been fermented. And then we will also have a control habitual diet as we eat in Sweden today. So fermented foods will be yogurt-like products, meat alternative, vegetables, kombucha, bread. So basically covering a portfolio of all types of foods that we eat, but select the fermented version of those foods basically. And the same corresponding foods, but non-fermented in the plant-based. And then in the control diet, the average Swedish diet. And as I mentioned, we will also do a stratification in this trial. So we will test 2 different microbial strata. So all these 3 treatments will be provided across 2 different microbial strata. And I will not tell you which microbial strata we will investigate, but we have a clear strategy for that. We will, of course, collect a lot of samples, so there will be stool sample. We are also considering to do meal challenge tests during the intervention. Blood samples and all the regular stuff will be also collected. And we hope that this study will be an extension of the Sonnenberg trial. Of course, our study is much larger, but not the least our design, where we have plant-based and corresponding plant-based foods which have been fermented, so we can disentangle the effect of fermentation. If we can talk about an effect of fermentation, a general effect of fermentation, because as I mentioned at the beginning, the foods are very different and they will provide very different effects and very different types of mechanisms. But at least the hypothesis is to test whether fermentation is good for cardiometabolic health in people who are also slightly perturbed in those aspects. I want to also mention that we have just finalized a study that we call Food for Gut Market. And in this study, we started up a literature review where we went through the literature to look for bacteria that are consistently associated positively or negatively with cardiometabolic risk factors. So we did a literature review of more than 8,000 papers, and we came out with these bacteria that are, of course, found by other reviews as well. That will be of importance for cardiometabolic risk factors. And then we did another review where we went through the literature for clinical trials that have tested foods, real foods, not supplements, and at least shown effects in either direction, consistent effects in either direction in at least 2 trials. And then we ferreted out those food items and used that as a basis for a food concept to address these bacteria that in step 1. And moreover, we also topped up with fermented foods. So we replaced, whenever we had a possibility to change an unfermented foods into corresponding fermented version, we did that. So we ended up with a dietary concept very rich in plant-based foods, not only plant-based foods, but from the literature that have been shown to affect these bacteria in either increasing that foods if they have been reducing that bacteria, or depleting that foods if they have been shown in the wrong direction related to the bacteria. And also adding fermented foods, in total 4 servings a day. to an otherwise diet which were in line with the Nordic food recommendations. And this diet was run in people. Half of them were BMI below 25, half of them above 25. And we recruited 40 individuals with signs of metabolic syndrome. Also, we screened and ended up with 30 completers. So they had 6-week dietary interventions on the concept diet versus a control, which is a habitual Swedish diet, and then they had a washout and another 6 weeks. The primary aim in this trial was to evaluate whether we could affect the gut microbiota composition for these critical bacteria, as I mentioned. And the secondary outcomes were whether we can affect the cardiometabolic risk factors, and also if we can mirror the changes of the potential changes of the microbiome in metabolites in the blood samples. And the expected findings is to see whether the intervention can alter specific microbes previously shown inversely associated with risk factors of cardiometabolic diseases, affected microbes if they will be linked with reduction of cardiometabolic risk factors, and also if this will be mirrored in the metabolome, as I mentioned, and also see if there is a difference in effects among those who are obese and non-obese, basically. And the idea was that we should have presented the data here today, but it's not ready yet. So I kindly ask you to stay tuned, and we will get back with results on this trial very soon to the community. So with that, I would like to end up with my take-home messages. So first of all, fermented foods is a very diverse group, many different microorganisms and very many different metabolites form. And of course, with such a diversity, we are targeting many different mechanisms at the same time. So it will be very difficult to find one effect of fermented foods. There will be many different effects, and the question is what the net effect will be at the end. For that, we need very characterized material that we are using in our trials and do very carefully controlled trials. Really, studies on non-dairy fermented foods like plant-based fermented foods and health, they are really lacking. So this is a big gap in the literature which we need to fill, and we are on the way to contribute at least partly to those gaps. And with that, I would like to thank you for your attention.
Thank you very much, Rickard, for this very nice and extensive presentation on health benefits. We go over to question and answers. There is not too much time anymore until 12 o'clock. However, we are able to extend our meeting slightly. There are some questions, so I assume that the presentations were rather clear from both Kristin and Rickard in that perspective. There is a question that can be addressed to both speakers, and that is about the short-chain fatty acids, which is of course, an effect of fermentation as such. However, the question is, do we have any evidence that the short-chain fatty acids that are already in the fermented products are also arriving in the colon, or are they absorbed already in an earlier stage? Who can I give the mic?
Kristin.
Okay, so those short-chain fatty acids that are already in the product do not make it into the colon. So they are absorbed in the proximal part of the gastrointestinal tract. But we do not know to what extent they reach the systemic circulation, whether they are also metabolized in the cells of the small intestine and so on. So these are things that we just do not know because it has never been tested.
Okay. Another question is about if we talk about the plant-based fermented products in comparison to the, for instance, the yogurt, the dairy-based fermented products. There is a problem with the anti-nutritional factors that we need to cope with, amongst others, phytic acid. So what is the role of fermentation in coping with anti-nutritional factors and increasing, for instance, bioavailability of the macro but also micronutrients? Kristin or Richard?
You can go.
Okay. You can decrease, if you decrease phytate by fermentation, which can be done, you can increase the bioavailability, but you have to prove that in humans, that it will increase the absorption of the nutrient and the nutrient status. And that is also something that has not yet happened so far.
So
I can add in there. We have done model studies where we, for instance, compare the bioavailability of iron and zinc when comparing faba bean protein, for instance, compared to fish and meat. And there are such studies also for other materials, and it's clearly shown. I mean, you can really. I mean, it's a very big difference, and I think there are also studies ongoing where they are comparing different processes of plant-based material and in a similar setting to give you really estimates of the bioavailability, the differences in bioavailability, at least across different processes in humans.
And did you test this also in humans? I was aware of in vitro digestibility tests that show that there is an increased availability, but not of human studies that really measure then how you affect.
We have tested with labeled material in a model, a human model system, comparing faba bean, the iron bioavailability with faba bean protein versus fish and dairy. It was published last year. I can send you the.
Have a look at it.
Sure.
Another issue with plant-based protein, plant-based fermented products is, of course, the protein issue. You indicated, Kristin, that the digestibility of plant-based protein is less. Is that increased in fermented products, and to what extent does the flow of the plant-based proteins may cause harmful effects instead of health effects if you look at proteolytic fermentation that takes place in the distal part of the colon?
Yeah, so you can increase the protein digestibility by fermenting the food up front, and then even, yeah, like with soy protein, you get to the same extent of digestibility as with animal protein. If you do not do that, so plant-based proteins are less fermented, that means that more protein reaches the colon where it gets fermented, where you have proteolytic fermentation, and that can have an effect on gut health. Now, we have done a study in the past in which we modulated the fermentation in healthy people and by changing the protein content in the food, so a high protein diet and a low protein diet. And then we saw that on the high protein diet there was more protein fermentation in the colon. That was, we could measure that from the concentration of paracrisol that were formed. But when we looked then at the toxicity of the fecal water, there was no difference. I mean, it was not the samples that had from the patients, from the participants on the high protein diet were not more toxic than the samples from the patients on a low protein diet. So which means that although these compounds are intrinsically toxic when you bring them in contact with cells, they were not important enough in the whole bunch of different chemical structures that are present in the luminal content to influence the toxicity there to indicate to, I mean, and to determine the toxicity of the colonic content. So it's difficult to estimate how relevant this proteolytic fermentation is for the toxicity and for the colonic health in humans.
Okay, thank you. And there is a question also about the technology of fermentation. Long fermentation process at low temperatures or short fermentation process at high temperatures. Is there any indication that this should matter, this could matter with respect to health benefits or digestibility or nutrient availability? So what is the interaction between the fermentation technology and the output? And maybe Christophe, you know a little bit about that as well.
I would just spontaneously say that we need to really look into what is the material, and also is it the compounds, what specific compounds we are addressing. I think that has to really be tailored to the right material and for the right target. I mean, exactly depending, it's too general to say that one or the other will have implications for health. If it's to reduce phytate, for instance, to liberate minerals, it would be one way, and if it's to form some other metabolites or degrade the matrix, that's another story, depending on what is the matrix. So basically, we need more information on this, and I hope that this project will contribute with that. I mean, to really study in detail and provide evidence for how these processes are affected by how the results of these processes, basically.
So there's little information available at this moment.
I think it's too little. I mean, to draw the conclusions about health, it's far too little, I would say. We really need to study that in detail.
I entirely agree. Yeah? So you have to look at it from on a case-to-case basis. That's one thing, and also from a food microbiome to food microbiome basis, because we have so many different microorganisms. The one has specific enzymes, proteolysis, the other has not. So it's too general, I think, to look at long temperature, long time, high temperature, short time, low temperature, whatever. It's not that easy, I think.
Yeah. Maybe to close the webinar with a nice question about fermented products, plant-based fermented products. What is the role of beer?
I think beer is a very interesting product. The only thing about it is that you can never make claims about health or anything when it comes down to alcoholic products. And that is where, of course, if you go to non-alcoholic beers or even low alcoholic beers, then that might change. And then you can start considering a fermented malt-based drink, let's put it then that way, as a carrier of healthy components like oligosaccharides, metabolites. So let's see what happens in the future there.
Thank you very much, and thank you all the attendances for joining us with this webinar. Thank you, Rickard. Thank you, Kristin. Thank you, Christophe, for presenting the project and presenting basics of gastrointestinal physiology and what is the status of the evidence of the health benefits of fermented products. Thank you, and see you later on in the next webinar. Goodbye.
Thank you.
Yeah.
Goodbye.
Bye.